The overall goal of this procedure is to monitor structural and functional alterations along the visual pathway in mice in vivo. This is accomplished first with an intravitreal injection of paramagnetic manganese ions, which serve intracellularly as MR-sensitive tracers and are applied as manganese chloride solution 24 hours before the scan. The second step is to arrange the anesthetized mouse inside the 3T MR scanner using appropriate hardware for image acquisition and monitoring of vital functions. Next, the mouse adopted and tracer-sensitive MEMR protocol is driven, and multiplanar 3D images of the intracerebral signal enhancement along the retinotectal pathway are reconstructed. The final step is to remove the mouse from the MRI scanner and to support its vital functions for rapid recovery from anesthesia, which allows for repetitive MRI studies of the same animal or subsequent behavioral or histological examinations. Ultimately, manganese-enhanced MRI of the retinotectal projection is used to show acute or progressive neuropathologies in experimentally or genetically modified mice mimicking neurodegenerative CNS diseases.
The main advantage of this technique over existing methods like postmortem histology is that structural alterations directly can be monitored in vivo or by repetitive studies on individual animals.
This method can help to answer key questions in the neuroscientific field, such as on structural pathologies in Alzheimer's or Parkinson's diseases, after CNS traumata, or even in poorly characterized metabolic diseases.
So this method can provide insights into special neuroanatomic features of the visual projection, but it can also be applied to